Hey everyone, this is chapter 7. This chapter is titled Energy for Cells and covers the process of cellular respiration. So cellular respiration is a process that allows for cells to make ATP in the mitochondria. So in the last chapter we talked about the production of glucose in the process of photosynthesis. So this process is going to be utilizing glucose that was made in photosynthesis to produce ATP. Cellular respiration requires oxygen and glucose, and it produces carbon dioxide and water. And this process is the reason why you breathe. We need to have oxygen, and thus we breathe in that oxygen every time we take a breath, and we breathe out carbon dioxide. So this is the reason for that gas exchange, is to fuel this process of cellular respiration. And essentially, cellular respiration is the opposite of photosynthesis. And so because they're opposites, we are able to utilize the reactants, the products of photosynthesis as the reactants of cellular respiration and vice versa. So if you look at the processes that are going on in this apple, it is a plant, it will perform photosynthesis, which if you recall from last chapter, produces glucose. That was the main goal of photosynthesis, was to take in the reactants and pump out glucose. Well, now our cells, or any cells that have a mitochondria, are capable of utilizing that glucose in order to make ATP. Just to give you a reminder of the general structure of a mitochondria, it has a double membrane, just like the chloroplast did in photosynthesis. The inner membrane is really convoluted. It has lots of ups and downs, that increases. You tell me. Think about it. The fact that it's not just smooth and has all these ups and downs, it is going to increase surface area. All right, so that those ups and downs, that inner membrane folds are called the cristae. All right, and of course it's found within our cells and inside of animal cells and plant cells. Pretty much any cells have mitochondria, but not all of them have not all of them have chloroplasts. All right. So in this process of cellular respiration, glucose is totally broken down. And that has to occur in phases. It breaks down slowly in multiple steps. It allows energy to be captured and to be used to make ATP. There's lots of coenzymes that are involved in this. Some of the important ones are NAD+ and FAD+. When they join with hydrogen atoms, we'll call them NADH and FADH2. But essentially, they function as electron carriers for an electron transport chain, similar to what we saw last chapter. Cellular respiration also involves redox reactions. And what that means is that in the reaction, there's oxidation that occurs and reduction. And oxidation is the removal of hydrogen atoms. Reduction is the addition of hydrogen atoms. So here is our equation for cellular respiration. And if you memorize your equation for photosynthesis, then this is going to be easy for you to memorize as well, because it's the opposite of it. All right, so cellular respiration starts with glucose plus 6 oxygens, and that yields 6 carbon dioxides, 6 waters, and energy. And that energy will be in the form, of course, of ATP. So in the oxidation part, it says oxidation is where we are going to remove hydrogens. The hydrogens are going to be removed from glucose. And those hydrogens will be used to form water instead after they're removed from glucose. In the reduction phases of cellular respiration, we will have addition of hydrogen atoms. So here you can see we have water that is going to be converted into oxygen that's going to be converted into water. Here we see then we have addition of hydrogen atoms. So oxidation, we removed hydrogens from glucose, and then oxygen, we added hydrogens to it. So this is a redox reaction. It has a reduction part and an oxidation part. Okay, so cellular respiration we said occurs in multiple phases. These are the phases of cellular respiration. Glycolysis, the prep reaction, citric acid cycle, and the electron transport chain. Now, some textbooks may call the citric acid cycle the Krebs cycle. And then some textbooks may also call the electron transport chain oxidative phosphorylation. Whoop. I might not have spelled that right, but I did the best I could. But just so you know, yeah, like I said, it might say in our textbook both, or it might just have one or the other. But unfortunately, sometimes in biology, we just have multiple words that mean the same thing. So again, glycolysis is the first phase. Then we have our prep reaction, the citric acid cycle, and the ETC. I'm going to refer to it as the ETC for short because that's just a lot easier for me to say. And again, here is our overall chemical equation. Glucose and oxygen will yield carbon dioxide, water, and energy. You need to memorize that. You need to know it for our test. So again, essentially what's happening here is glucose in the presence of oxygen is being broken down into these components here. And it happens in phases. The first phase, like we said, is glycolysis. Glycolysis is where we break down glucose into the first intermediate, which is pyruvate. And in glycolysis, we have simultaneous production of ATP, just a little bit, but it still does produce some. This phase occurs in the cytoplasm. All right, you need to know that as well. It occurs in the cytoplasm. So we haven't made it to the mitochondria yet. Now, glycolysis happens in two steps. One is the energy investment step, and the other is the energy harvesting step. So essentially, 2 ATP are used to activate glucose, and then as a result of its activation and its ability to perform glycolysis, we're able to harvest 4 ATPs total. So if you do the math, then there's a 2 ATP net gain. Like, for example, if I were to give you $2 and you gave me back $4, then I would have gained a net gain of $2. So same thing. We invested 2 ATPs because it's kind of like the molecular currency of cells. It's molecular money. We invested 2 ATPs. We got out 4. So we have a net gain of 2 energy molecules, ATP. In glycolysis, we also have NADH being produced. It's used later as an electron carrier. And you might be wondering, well, how is it an electron carrier? And I believe we talked about this in the last chapter, but I'll go over it again. So this hydrogen here is an electron carrier because what is a hydrogen atom? Think about it. I'm going to draw the chemical structure here. So here we have the nucleus of our cell. I'm going to write H for hydrogen. So what is hydrogen? It's a proton in the nucleus, and it's a single electron. This electron here is what I'm referring to when I say that NAD plus was an electron carrier, because it can have attached to it that hydrogen, which can lose this electron and because it can ionize, form an ion, and that is how it can be utilized as an electron carrier. That electron can then be passed down an electron transport chain that happens later. All right, so again, 2 electron carriers, NADH, are produced here from NAD+, and they'll be used later. Now, this process of glycolysis is anaerobic. That means it doesn't need oxygen. Overall, cellular respiration is typically going to be completed in an aerobic environment, though. You need to know the difference. Aerobic versus anaerobic. All right, aerobic means has oxygen. Anaerobic means no oxygen. So glycolysis doesn't need to have oxygen present in order for it to occur. And then next, one 6-carbon glucose molecule is broken down into two 3-carbon molecules, and they are called pyruvate. So I included this picture over here because, like I've told y'all before, I'm a picture person, and this really helps me to visualize and remember the inputs and outputs. So in glycolysis, of course, we had an input of glucose. 6C glucose just means it has 6 carbons. We know that, right? Because we said C6H12O6 is the chemical formula for glucose. So we have our 6-carbon glucose, 2 NAD pluses, 2 ATPs, because of course that's the 2 we are investing, and then we also have 4 ADPs and 4 phosphate groups. We're going to have those in order to form ATP by adding these 4 phosphates to these 4 ADPs to get their total of our 4 ATPs total. Okay, so our outputs are listed here. We're going to have an output of 2 pyruvates, which are 3-carbon molecules because we broke it down from glucose. So you can think of it like this. So each one of these little balls here are are carbons of glucose. If we break it down, split it in half, now we have 2 3-carbon molecules, and those are pyruvates. Our NAD pluses have a hydrogen atom attached to them to form 2 NADHs. And then our 2 ATPs that we invested will generate a total of 4 ATPs total. So we have a net gain then of 2 ATPs as well as these items as outputs. Okay, so that is glycolysis. Now in the next step of cellular respiration, those pyruvates are going to move into the next phase. And different things will happen to these pyruvate molecules depending on whether their environment of the cell is anaerobic or aerobic. Again, anaerobic means no oxygen, and aerobic means there is oxygen. So we're going to go to the right side of this first. We're going to talk about aerobic respiration first. So again, we started off with glucose. It went into glycolysis. We yielded pyruvate and ATP. And now that pyruvate is going to move into the next phase, which is aerobic respiration if oxygen is present. And that's where the next phases of cellular respiration will have in the mitochondria in an aerobic environment. All right, and as a result, we're going to produce carbon dioxide, water, and ATP, specifically around 36 to 38 of them. Okay, so if oxygen is available, those 2 pyruvate molecules from glycolysis will enter the mitochondria, and they will be paired with 2 coenzyme A's. That will cause pyruvate to be transformed into another molecule called acetyl-CoAs. And carbon dioxide will be yielded as a byproduct. Again, this is our prep reaction. We have 2 pyruvates. They're coming from glycolysis. They will bind with, they will have a chemical reaction with 2 coenzymes A, and that will yield 2 acetyl-CoA. And then we have carbon dioxide as a byproduct. We will breathe out that carbon dioxide. All right. So you might be wondering now, because I've also listed our overall equation for cellular respiration over here. Well, where's pyruvate in this equation? And the reason why pyruvate is not listed is because anything that is an intermediate is not going to be listed in that chemical reaction because it's going to be used to make something else. So one thing you do see is glucose. That was our very first input. We don't see pyruvate because pyruvate is going to be the reactant of our next equation, of our next phase of cellular respiration. We do see carbon dioxide though, because we're done. This carbon dioxide is being breathed out. It's done. And we only have 2 of them. There will be a total of 6 after all is said and done. Okay, the next part of cellular respiration is the citric acid cycle or the Krebs cycle. So you can see the beginning of the Krebs cycle. What do you know? We have our 2-acetyl-CoAs. They're going to be what is entering into this. Again, therefore it's an intermediate. It's that product that we got in our previous step to get the acetyl-CoAs is now the reactant in the next step in the next phase. That's why it's not listed in our overall equation. So this part of our cellular respiration process occurs in the matrix of the mitochondria. Remember, the matrix is the fluid, and it's a circular pathway. You can see it has lots of steps and products and byproducts, and I do not expect you to remember or memorize all of these little details here. But what I do want you to know is that acetyl-CoA is the input here. I do want you to know that. Each turn of this cycle produces carbon dioxide. You can see carbon dioxide here, for example. Carbon dioxide over here. All right, again, there's our it's done. We're done with those carbon dioxides. We're going to breathe them out. They're not going to be used in another step, so they are listed in this equation. Now, this process is going to occur twice per glucose molecule. I want you to know that, and I want you to know why, to ensure that you're truly understanding all of this stuff. So why does this cycle turn twice for every glucose molecule that we consume? Well, when we broke down glucose in glycolysis, it yielded 2 pyruvates. Those 2 pyruvates then made 2 acetyl-CoAs. So for every acetyl-CoA, it enters this cycle. So we had 2 acetyl-CoAs from the breakdown of each glucose. So that's why this cycle turns twice for every glucose molecule we have. All right, and here's our inputs and outputs. So we have 2 acetyl-CoAs that entered in. We also have NAD plus and FAD. I'm not really too worried about you memorizing this part because we know that they're just electron carriers, and we'll get to those later. But you do need to know that acetyl A comes in here. We have an output of some carbon dioxide and a little bit of ATP. Next, the ETC. So this occurs in the cristae. That's the inner membrane that is highly convoluted in here. It has all these ups and downs. Excuse me. That is where the ETC occurs, which makes sense, right? Because just like in photosynthesis, we had the electron transport chain occurring in the thylakoid membranes, which is a phospholipid bilayer, just like the cell membrane. So same thing here. The cristae are made up of a phospholipid bilayer. Okay, so this image here shows you the electron transport chain that is in the cristae. This is the inner membrane space on this side, and this is the mitochondrial matrix on the other side. And of course, you see our phospholipid bilayer with several different proteins in blue. And so what happens here in the electron transport chain, all of these different NADHs and the FADH2s that were our electron carriers are going to pass electrons down this chain of proteins. So the electron stems from NADH or FADH2, which is because they have hydrogens, and if you break down a hydrogen and allow it to ionize, then that's going to yield a hydrogen ion plus a free electron, just like I drew a couple slides back Again, this is our hydrogen atom. It has a single electron. If it loses that electron, then it becomes positively charged. So we have a hydrogen ion. All right, and so that's what happens when NADH breaks down, or FADH2. It releases these hydrogen ions with these free electrons that can then be used to be passed down the electron transport chain. So as the electrons are passed down this chain, hydrogen ions are going to be pumped across our inner mitochondrial membrane. Right, so you can see that here. The little lines being drawn showing hydrogen ions going on the other side. All right, so that probably sounds pretty familiar, I hope, because we talked about it in photosynthesis. That's going to cause a hydrogen ion electrochemical gradient because you're going to have lots of hydrogens on this side of the membrane, but relatively few over here. And the cell doesn't like that. That has potential energy there. The cells are trying to reach equilibrium. That's their goal. But because they're pumping all these hydrogen ions onto one side, it's causing a buildup of potential energy. So those hydrogen ions then are going to diffuse through ATP synthase, just like we saw in photosynthesis. And for every hydrogen ion that passes down, ATP will form from ADP and the phosphate group regenerating. So for every glucose molecule that you consume, there's about 32 to 34 ATPs that will form from that consumption of a single glucose molecule. At the end of the protein chain, the electrons are accepted by oxygen, and that allows for the formation of water. All right, and you'll see that here. So electrons are passed down this chain, and they meet up with oxygen, which then allows for the formation of water. That will then be excreted out of the body. My handwriting is terrible, y'all. Excreted. Okay, so this shows you all of the different proteins that are inside of the inner membrane of our ETC. All right, so in total, from glycolysis, we got 2 ATPs. From the citric acid cycle, we got 2. And there's 32 to 34-ish that stem from the electron transport chain. And so therefore we have a total of 36 to 38 ATPs that is going to be generated from a single glucose molecule. You need to know that. All right. Uh-oh. That's not good. Okay, sorry about that. So next, energy content. Something I want you to think about that we've discussed before about how transformations of energy are never perfect and they result in a loss of energy. So our reactant of cellular respiration, which is glucose, contains about 686 kilocalories worth of energy. But the energy yield, 36 ATP, is only about 263 kilocalories worth of energy. That's a huge reduction here, 263 versus 686. That's telling us then that the efficiency of cellular respiration is only about 39% efficient when it converts glucose to ATP. The rest of that energy is lost as heat. Second law of thermodynamics. Okay, so next is fermentation. Fermentation is what occurs in an anaerobic environment. So if our cells are depleted of oxygen, then the pathway will go into fermentation rather than aerobic cellular respiration. All right, so that's when oxygen is not present. So this process of fermentation occurs differently depending on the type of organism we're referring to. In animals and some bacteria and fungi, we will have lactic acid fermentation. It's called lactic acid fermentation because it results in lactic acid being formed as well as ATP. And then in plants and yeast, ATP will form as well as CO2 and some sort of alcohol. So we'll discuss animals first. So whenever I say that our cells are depleted of oxygen, it doesn't mean that, we're about to die and because of course we can't live without oxygen. It just means that our cells are running out of it and it causes an oxygen deficit. We're still able to breathe and get in more oxygen, but our cells are just tired. So in lactic acid fermentation, we're going to have regenerations of NAD plus from NADH that can then be reused in glycolysis. This was done in the ETC when oxygen was present, but in this case it's not, so it will be regenerated in glycolysis. So glycolysis can continue over and over again. And then pyruvate, we reduce pyruvate from glycolysis to lactate. So it's different here too. Lactate and lactic acid are the same thing. And when that builds up in our muscles, it causes them to feel tired and sore. So if you've ever been to the gym before and your muscles were sore and tender the next day, that's due to lactic acid buildup. And then also as a product of lactic acid fermentation, we have a little bit of ATP. So in plants and yeast, we have alcohol fermentation. So in this process, pyruvate is going to In this process, pyruvate is going to be regenerated as well, and NAD plus is going to form back to NADH for reuse during glycolysis. Again, this was done in the ETC if oxygen was present, and we have a production of carbon dioxide and ethanol or some sort of alcohol. So this is why plants and yeast can be utilized to produce alcohol. In beverages, for example, yeast are used in the production of beer. We have grapes, of course, that can be used to help in the production of wines, and various different plants can be utilized to produce other alcohols. Now, there's some advantages and disadvantages to fermentation. And one advantage is that it provides a quick burst of ATP. This process is much faster than going through all of the different pathways of aerobic cellular respiration. But there's a lot of disadvantages. So lactate and alcohol are toxic to cells. And it's kind of funny because it's even possible for some organisms, yeast, for example, that make alcohol, can actually die from the alcohol that they're producing. Also, lactate changes the pH of your cells. It causes our muscles to feel tired and sore because of that oxygen debt. It's very poorly efficient. This is only about 2.1% of efficiency of breaking down glucose into ATP. However, of course, it's when our muscles are in desperate need of ATP that they're going to do this, and they don't have any oxygen. They still need ATP, so it allows for that quick burst. It's just not very efficient. So, of course, our cells are going to prefer to use aerobic cellular respiration, but in cases where that's not possible, this is kind of a last-ditch effort to get you some ATP. Okay, so something to think about. Whenever we eat food, typically it's not a whole bunch of glucose. Think of a turkey sandwich. We're going to have proteins in the meat. We're going to have carbohydrates in the bread. We're going to have fats in the cheese. So all of those different things are going to be able to play a part in cellular respiration, not just the glucose. So this shows you that metabolic pathways are not closed systems. So lots of these different things can be utilized in some way, shape, or form to make ATP. So, for example, carbohydrates, amino acids, and glycerol, which are one of the components of fats, can be used to fuel the fire for glycolysis. Fatty acids and amino acids can help with pyruvate oxidation, which is the prep reaction. Amino acids can be used to fuel the fire of the citric acid cycle. Right, so even fructose, galactose are sugars that can feed into glycolysis. Glucose is our body's preferred mechanism which is the way our textbook teaches about it, but that doesn't mean that other things cannot be utilized to help. This shows you the exact same thing. It just shows you in the form of a pizza. And I just really like this picture, so I wanted to incorporate it. But it shows you that the proteins from the meat on the pizza are going to be broken down to amino acids. They can be used to help with the formation of pyruvate and acetyl-CoA and the citric acid cycle. Glycolysis, like we said, is our preferred method. We have fats as well that come from in pizza, and they can be broken down to glycerol and fatty acids, just like we talked about in chapter, I believe that was 2 or 3. But that glycerol can help with the process of glycolysis. It can also help with the formation of acetyl-CoA in the prep reaction. So all of these different nutrients we eat are going to be components of our metabolic pool, and we can utilize those components to fuel all of these different phases of cellular respiration. All right, you guys, hope you enjoyed it.